you very much to be with us today. It's an honor for the Carlos Chagas Filho Institute of Biophysics from the Federal University of Rio de Janeiro, and especially for the graduate program in biophysics to have you as a speaker in the discipline of our program. We have a lot of students listen to you today, and for us it's a big pleasure to have to you like a speaker. Please feel at home and be very welcome to the Carlos Chagas Filho Biophysical Institute. Thank you very much. Professor Vanderlei.
Okay, John, thank you very much to accept our invitation to participate of this web symposium. That's a different way. We would prefer to have you here, but since you come to Brazil quite often, we will in the future invite you to come here. Professor John Kelly comes originally from the University of Glasgow, where he did all his basic work, the undergraduate courses, and he got the PhD grade at Glasgow University in 1978. In 1980, I had the opportunity to stay at Glasgow University for a few months with my old friend Kit Vickerma, where we did some work together with Steve Hadjuk, Lawrence Tetley, David Barry, probably people that you know very well. But after Glasgow, he made a postdoc at the Cancer Research Center in London, and then entered at the famous and well-known and well-recognized London School of Hygiene and Tropical Medicine, where he did all his academic career from the basic to the top, and now it's one of the most distinguished cell biologists or parasitologists to put the things together and have given an important contribution to a better understanding of Trypanosoma cruzi, its interaction with the host, using new approaches in bioimage. So that's he has also worked quite intensely in the area of chemotherapy, also against Chagas disease, look for the mechanisms of drug action and resistance, understanding the pathogenesis of Trypanosoma cruzi infection, and exploit in vivo image technology to study diseases, pathogenesis, and assess drug effectiveness. I believe, John, that after this talk, probably we'll make another appointment to discuss possible collaborations, because I think that we can do several things together. So that you can.
Okay, I can start. Thank you very much, Vanderlei, for the invitation. And again, I think the people who have put together this symposium in the very difficult circumstances we have at the moment have done a fantastic job, and hopefully I can contribute to it. So today I want to essentially go over some of the work we've been doing on Trypanosoma cruzi and the pathogenesis of Chagas disease over the last few years using mouse models. And I want to give more of an overview and rather than dig down too much into the minutiae, and hopefully it will be more understandable to maybe a wider audience. So in terms of that wider audience, I'll just spend the first couple of slides giving some background into Chagas disease and the parasite, and this will form the basis of some of the experimental data that I will subsequently include. As I'm sure most people in Brazil know, Chagas disease is the main parasitic infection in the Americas. For those who don't know it too well, it infects a very large number of people, 5 to 10 million people, predominantly in Latin America, but more recently through migration, then there are large numbers of infected individuals in North America and in Europe, including here in London. The disease is caused by the protozoan parasite Trypanosoma cruzi, which is transmitted predominantly by triatomine bugs, one of which you can see on the screen here. And other routes of transmission include the congenital and oral routes, and these are increasingly important. So let me go on now to just describe the disease, because I think that's important for understanding some of the data that I will be presenting. And traditionally, the disease has been divided into 3 stages, although there's not seamless transitions between each of them. In the acute phase, which follows after infection, in humans, this can be between 1 and 8 weeks post-infection. Then the parasite is widely disseminated and present in relatively high numbers and can be easily detected in the bloodstream. There is a 5% fatality rate in diagnosed cases. I sort of emphasize diagnosed because the vast majority of cases are not diagnosed as such, because the acute phase is generally a sort of mild febrile illness that often passes unrecognized. So most people do not realize that they've been infected, and that is one of the issues. Following the adaptive immune system kicking in and reducing significantly the parasite burden, and I'll show you some slides on this in a minute, patients then go into what's called the asymptomatic chronic phase, which for the majority, or at least 70% of cases, lasts for the rest of their life. I should emphasize this is a lifelong infection. However, the major pathology associated with the disease is in a group of about 30% of patients who develop the chronic pathology. And this often takes decades to become manifest, and there's no effective cure for these pathologies. The major pathology associated with Chagas disease is to do with the heart, and I'm showing it on the screen there, the effect of cardiomyopathy. And there's a number of heart conditions that follow from infection with T. cruzi, and it's probably the largest infectious heart condition in the world. Other symptoms include the megacolon or the so-called digestive tract megasyndromes that can occur in some other patients. So we don't really understand why only 30% of the patients develop the chronic pathology, and we don't understand why some develop cardio pathology and others develop the digestive problems. These are big questions that are still remarkably unanswered. Okay, so let me now push ahead and ask how we've been tackling this problem. As you can guess from the complexity of the disease and the long-term nature, it's difficult to do experimental science with human patients. And so animal models have always been a mainstay of T. cruzi research. And we've been developing bioluminescence imaging to monitor disease progression in mice. And this has involved the genetic modification of the parasite to introduce a red-shifted luciferase gene that emits light at the red end of the spectrum. And at the red end of the spectrum, you have greater tissue penetration. So the general way we undertake these experiments is to inoculate the mice with the parasites, and at various points thereafter, we inject with luciferin. Luciferin is the same substrate used in fireflies to generate bioluminescence in there. So we then anesthetize the mouse, acquire the image, and then the animal's returned to its cage. And under our animal license, we can do this 40 times in the lifetime of a mouse. And on the upper screen there, we show a mouse that's been followed for over a year. And what you can see is the typical heat map model, the heat map that you get with bioluminescence imaging. And at day 14 of the infection here, we're at the peak of the acute stage in the mouse, where there's a high parasite burden, but the adaptive immune system then reduces that significantly, And by the time we get out to day 50 to day 70, there's a transition in then to the chronic infection that lasts the lifetime of the mouse. Now, the heat maps here, I should emphasize, are on a logarithmic scale, so that at the peak of the acute stage, for example, there are possibly 1,000 times more parasites than there are during the chronic stage of the infection. Okay, so let me now progress. So one of the first questions we sought to address using the bioluminescent imaging, and again, trying to understand the patterns and the profile of the infection and how the pathology develops. We try to ask, whereabouts does the parasite persist in the chronic stages of the disease? And this has always been a difficult problem to address because the parasite burden is so extremely low. There's so few parasites in a mouse and similarly in humans. And the bioluminescence imaging system allowed us to do that. So let's look first at the acute stage where there's less technical issues. And as expected, in the acute stage, the parasites are widely disseminated. And you can see in the organs that are laid out in the presence of luciferin here, that all of the organs are highly infected. So the infection is pantropic at the acute stage of a disease. And you get all organs and tissues infected. Now, on the right-hand side of the screen here, we've used a modified, construct in which we have joined together the firefly luciferase and a green fluorescent protein called M-neon. And this expresses a fusion protein in the parasite that is both bioluminescent and fluorescent. So this allows us then to use bioluminescence to identify infected tissue, and then we can use histological sectioning and confocal microscopy and identify the parasites on the basis of fluorescence. So what you can see here on the right there is a number of different organs and tissues from the acute stage mouse, and as you can see, they are all relatively highly infected. Now, the situation in the chronic stage is different. In the chronic stage, certainly in the mouse models that we've been working with, the GI tract seems to be a privileged site for parasite persistence. This is where the parasite survives in the chronic stage of the infection. And again, remember, this is for the life of the parasite, of the host. So what we have here on the screen is 3 mouse models, each infected with one of 2 strains of T. cruzi. Strain, the lineage 6 is the C.L. Brenner strain, and then the lineage 1 is one of the common lineages that we find with this particular DTU. And again, what you can see there is that the colon is a predominant site of infection as well as the stomach. However, what you can also see is that this restriction to the GI tract is not complete, and that there are variations in mouse models. In the C3H, for example, you can see wider dissemination of the parasite into other organs and tissues. So the C3H mouse certainly doesn't restrict the parasites so definitively to the GI tract, although in practice, none of the mice do completely. Now, the first thing that surprised us when we saw these data originally was, why isn't the heart infected? The heart is the main site of pathology. Well, the heart is infected, but it's not infected all the time. So remember, what you're looking at here is snapshots of the infection. In a bulb C mouse, for example, probably 10% of mice that we look at will be infected in the heart at the point of sacrifice. In C3Hs, the level is 70% or so will show heart infection at any one point in time. So how does this relate then to pathology of the disease? Because the pathology is down to cardiac issues. And what we found is that cardiac pathology develops regardless of host parasite genotype combination. So for example, with the bulb C mouse on the top there, you get the pathology. And pathology here, we're using fibrosis as a proxy for pathology. And we're looking at collagen deposition, which stains blue, and we can use multiple sections and software on the imaging system to look at the extent of this blue staining to give us a measure of fibrosis. And what you see is that fibrosis occurs in all of the mice strains, even though they don't all constrain the parasite to the GI tract to the same extent. Again, if you look at that now on a table, again, you can see on the cardiac fibrosis score here compared to non-infected mice. You can see significant levels of fibrosis in the biopsy, this C57 black, that's a B6 mouse, and to a slightly greater extent, the C3H mouse. So the important take-home message from that is that cardiac pathology can occur in the absence of locally persistent infection. So I think it's important to think about, if I can just go back a slide actually, think about tissue tropism in the context of Chagas disease. Our feeling is that tissue tropism in Chagas disease is not a specific property of the parasite in terms of how it specifically migrates to organs and tissues. Rather, tissue tropism is determined by the immune system, we would predict, and that the parasites are present in the gut simply because that's one place where they're not readily killed, and they're absent from other sites because they're destroyed by the immune system. The tropism is not an active process in Chagas disease, or at least that's the way it looks. Another thing that surprises was the dynamic, highly dynamic nature of infection, because if you read in any of the textbooks, which I'm sure a lot of the students have done, many of the textbooks will tell you that essentially Chagas disease is it results from a latent infection over many years. But as you can see from the slide here, it's anything but latent, and the infection is highly dynamic. So what we have here is a mouse that from day 125 of infection, a ventral and dorsal image of the same mouse that we followed for 2 days, and we've imaged at various points. So for example, here you can see two bioluminescent foci that correspond to infections, and you see that over time these gradually disappear, so that by the time we get to 24 hours later, these foci have disappeared. Elsewhere, if we look at here, you see a focus that appears, then disappears. So this is a highly infectious, highly dynamic infection foci that are appearing and disappearing over a period of hours. And it was certainly very puzzling to us why the infection would take this type of profile. Another thing that we found more recently is that the skin is also a site of parasite persistence. This is something we probably didn't recognize at first, because again, in most cases, you image the mouse from the outside, obviously. However, when we sort of refined our techniques and we took the entire skin and we laid it fur side down in luciferin, you see here that there is a high number of mainly quite small infection foci in the skin of, in this case, 2 mouse strains. These infections are highly variable from mouse to mouse. And again, the foci themselves go from very faint to quite detailed. And we can actually look at that in more detail, again, using the bioluminescent fluorescent protein. And that allowed us to pick up, in this case here, an infected cell very close to the surface of the mouse, about 150 microns. And obviously infections at this site in the mouse will be important for transmission. So that's the skin, but let's look at the wider context of infection. And I put up here a number of relative levels of infection in various muscles, in various tissues, including skeletal muscle, the gastrointestinal tract, and some of the internal organs. And what you can see with bowel C mice, for example, then you commonly find infections of the GI tract, rarely of the skeletal muscle, and rarely of the heart. However, above the dotted line, which is 2 standard deviations in bioluminescence above the background, you see that with C3H, again, because the infection is less restricted to the GI tract, you see infections occurring more frequently in the heart, but also in the skeletal muscle. And in this mouse strain, the skeletal muscle seems also to be a site where we often find parasites. And that's shown here. So this is the skeletal muscle that we're looking at now. And what you can see here are the red is the staining with actin, and the green are the parasites. So you can see here there's a huge parasite nest. We call these mega nests that contain hundreds, if not thousands, of individual parasites. And in the C3H strain, but not bulb C, this is quite common to find 1 or 2 of these per mouse. Okay. So that's the location of the parasites in the mouse. What about the parasites themselves? What's the nature of the parasites in the gut reservoir? Why are they able to survive? Is there something about the nature of the parasites that allows them to evade the immune response? Or is there something about the replication of the parasites that is different from, say, in the acute stage? And one of the first points I want to emphasize is, again, talking about this restriction of parasites to the GI tract, is that it's immune mediated. And that's shown in this next experiment, where we've taken a chronically infected mouse, and we've then treated them with cyclophosphamide, which is an immunosuppressant drug that knocks down the level of B cells and T cells. And what you can see in this mouse is, over a period of days, an increase in the parasite burden following immunosuppression. And by the time we get to days 13 and 16 after immunosuppression, you can see that the parasite levels are back up to the levels they were in the acute stage. So that's a 100 to 1,000-fold increase in the parasite burden during this period. If we look at the internal organs, again, we see a similar pattern at day 0. That's during the chronic stage of the disease. You see parasites in the colon, you see some in the gut mesentery tissue, some in the stomach. However, 6 days after the initiation of immunosuppression, you see that parasites are now beginning to amplify in the GI tract. And by day 9, there's wider dissemination of the parasite burden. Now, we can't say definitively that parasites are spreading from the GI tract, but certainly, I think this is fairly strong evidence that it's the immune system that restricts parasites to the GI tract and prevents them from infecting other tissues so that's the reason that they persist, not because they target the GI tract, simply because they are eliminated very rapidly at other sites. Now, I'm sure you're looking at these images here. How many parasites do these blue dots correspond to? And that was something that we couldn't really address very easily at first. We knew when we did in vivo imaging, that's looking at a whole infected mouse, then the limit of detection was between 100 and 1,000 parasites. But we couldn't really do this very easily with the internal organs using this ex vivo imaging system that I'm showing here with the organs. And it's only recently that we've been able to establish a limit of detection in terms of picking up the number of parasites with bioluminescence imaging. And this is all down to improvements in how we process the organs. So what we've got here is a strip of tissue from the colon of an infected mouse. And you see on the left-hand side of the screen here, there is an infection foci around here. And we then excised that region and undertook serial imaging in sections. And what I'm showing you in the middle image here is 2 sections. The lower one relates to the larger bioluminescence foci, and the upper section here to this very small foci here. And we estimate, we calculate that there are actually 16 parasites in this small focus. So that would be the limit of detection. Okay, it doesn't look like the 16 parasites in this section here, but this is a two-dimensional image of a three-dimensional structure. So to calculate more accurately the number of parasites, we use a process called Zed Stacking, which we can use with confocal microscopy. And with Zed Stacking, we take images sequentially through the tissue section, and that allows us then to create a three-dimensional model for the infected tissue, and allows us accurately then to define the number of parasites. Here I've shown blue staining here, which identifies DNA, and you can see here the small dots there that correspond to the individual parasites in this infected cell. So on the basis of this, I was actually surprised how sensitive the bioluminescence imaging can be, and it allows us then to look at the entire colon of animals and try to identify more definitively the number of parasites, where they're localized, and the cell type in which they are located. And let me just now go through this next slide bit by bit. What we found, and I'll give the take-home message first, is that the majority of parasites in an infected colon are located in the circular muscle layer. So there's a cartoon there that shows you the structure of the colon, the smooth muscle, the mucosal layers, and the epithelial cells. In the middle image here, we have a phase image, and one on the right-hand side, the same tissue stained with DNA. And on the lower graphs here, we have then the percentage of infected cells and where they're found. And you can see that in two mouse models, approximately 60 to 80% of the parasites were found in the circular muscular layer. Very few in the mucosal or the longitudinal layer. And again, that's the type of image that we analyze when we take these sequential histological sections and examine them, looking for green fluorescence, and that allows us to go through the entire colon of a mouse to try and estimate the precise number of infection foci and the number of parasites. Okay, so which cells are infected? Well, it appears that it's mainly smooth muscle cells within the muscle, within the muscular layers that are the main infected cell type. And that's shown in the upper image here, where we've got an antibody specific to smooth muscle cells. And you can see here, these are the parasites in here. They appear yellow simply because they're expressing a green fluorescence on a red background, so they appear yellow in that context. Below here, we have a beta tubulin that's specific for neuronal tissue, and you see that around about 10% of the parasites in the colon are infecting neuronal cells in that locality, and a very, very small number in infecting nucleated hemopoietic cells. Okay, so what about the parasite numbers? Well, again, this was something quite a little bit of a surprise for us, because in the past it's always been very, very difficult to detect infected cells during a chronic infection, because the parasite load contain more than 200 parasites. So what you see in this upper graph here is individual infected cells. This is 200 parasites, and you see here that some of the infected cells contain over 1,000 parasites. However, in the chronic stage, it's very rare to find infected cells that are contain more than 100 parasites. Generally, the acute stage infection, the result is that parasites escape from the host cell before they get to very low levels. Again, I should emphasize that although the majority of parasites are present in host cells containing more than 200, then the majority of cells that are infected contain many less parasites for that. And that's shown in the lower graph here, where we look at the percentage of parasites relative to the nest size. So that although there are a lot of host cells that are infected with small numbers of parasites, it's those relatively small number of host cells that contain a large number of parasites that contain the majority of the parasite population. And again, this is just going back to showing you how we estimate these numbers. What I'm showing you here now is a piece of colon tissue that's been excised here, and we've then imaged it with a confocal microscope, and you can see the green parasites, and here you see the blue staining of the DNA. So how we calculate the parasites is shown down here. In this, we take a number of serial Zed stacks, and we count parasites based on the kinetoplast DNA. It's a mitochondrial DNA, because the mitochondrial DNA is much more concentrated, and it comes out as a very easily detectable blue bar, whereas the nucleus tends to be more diffuse. And that allows us to get quite accurate estimations of the number of parasites in an infected cell. Another example of a highly infected cell is shown on the right here. This is a massive infection in the colon of a chronically infected mouse. This is right at the end of the infection, because you can see here that the infectious trypanosomes are beginning to escape from the host cell. So I would think that very soon in this particular animal, if it hadn't been sacrificed, all of these parasites would have escaped into the systemic system. Okay, so what about the replication status of parasites? Well, I think we were, again, quite surprised to find that replication inside a single infected cell is asynchronous. And we established that using two approaches. First was using this thymidine analog, EDU, ethinyl 2-deoxyuridine. And when you inject an infected mouse, EDU gets incorporated into replicating DNA. And using click chemistry, you can then detect which parasites or which mouse cells are in S phase, so replicating the DNA at the point of injection. Now, thymidine analogs do not survive very long in the bloodstream or tissues. They're bioavailable for approximately 15 minutes. So what this technique provides is a snapshot at that particular time of either host cells or parasite cells that are undergoing replication. And an example of that is shown in the bottom left here, where we've got adipose tissue from an acute stage infection. So here we have the green parasites, the DNA of the parasites, and in the EDU labeling here, you can see that some of the parasites label with EDNEU and some do not. And that simply says that within this one infected cell, some parasites are replicating the DNA and some are not. This is another example from chronic stage infection. Here we have one of these huge mega nests in the colon, and we've taken serial sections by histology and then looked at the staining of the parasites. And again, right through this huge nest, you can see that some of the parasites are replicating, the majority, however, are not. So there doesn't seem to be any programmed replication going on in infected cells. And at least by EDU labeling, that would suggest it's an asynchronous process. Whether it's stochastic or not, we don't know yet. Another approach to looking at replication is to use what are called tunnel assays. And tunnel assays allow us to specifically identify kinetoplast DNA, that's the mitochondrial DNA that's replicating. And the unusual nature of the mitochondrial DNA in trypanosomes means that there are hundreds and thousands of these small mini-circle DNA molecules present in the mitochondria. And the process by which they replicate results in transient nicking of the DNA, that's breaks in the DNA, that can then be labeled by terminal transferase, which requires a free 3,5-hydroxyl group. And you can use labeled UTP to identify replicating kinetoplasts. And that's shown on this slide here, where we have tissue excuse me, from a stomach, from a chronically infected mouse. Again, you can see the green parasites, the DNA staining of the parasites. And again, when you look here at the merging of the tunnel and the DNA, you see that some of the parasites are replicating DNA and others are not. Therefore, on the basis of replication of mitochondrial DNA and nuclear DNA, we can say that replication of the parasite seems to be asynchronous in individual host cells. So what about replication rate? This is a question we approached using the experiment outlined here. So we've got 3 groups of mice: an acutely infected mouse and 2 chronically infected mice. Here, what we've done is inject them twice with EDU in one day. So we inject them 6 hours apart with EDU, and then the following day we sacrifice. So we've done that for the acute stage mouse and the chronic stage mouse. And then for the final group of chronically infected mice, we injected them twice a day for 3 days, once finally on the 4th day, and then we sacrificed the mice. And the outcome of that is that we get pictures like this that I've shown you before, where we get parasite-infected cells in which some of the parasites are stained with EDU. That indicates that they're in S phase at the point that they were exposed to the thymidine analog, and they're replicating their DNA. So what we found, and on the left of the screen here, I simply get a table to show you the large number of cells and infected cells and parasites that we analyzed from an acute stage and chronic stage. This is a 1-day infection. And what you can see here is that in the acute stage, using this process, we label approximately 60 to 70% of the parasites with these 2 injections 6 hours apart, whereas in the chronic stage, the number of cells that are EDU positive is around about 20%. So that's more than 3-fold difference. So we're looking, again, at snapshots of parasites that are replicating their DNA. And I think by inference, you can then take it that parasites in the acute stage are probably replicating faster than they are in the chronic stage. When we did multiple injections, that's in the 3rd group here, you can see that we can get, if we give us a total of 7 injections over 3.5 days, we get the number of labeled parasites up to 40 to 50%. So that tells us that labeling is cumulative in the chronic stage. But even with this extended period of labeling, we're not yet at the level that we got with 1-day acute stage infection. And that just, again, identifies the in a bar chart form. Okay, so it appears that parasites replicate slower in the chronic stage than they do in the acute stage. I presume it's because there are signals in the environment that they recognize, well, this could be in the acute stage, they replicate faster. And we can also see from this bar chart that as the parasite number in a host cell increases, there seems to be a slight increase in the replication rate of those parasites. And that, again, here's just some more examples of that to show that we analyzed huge numbers of these images, and I just given you 3 images there from a 1-day labeling acute and chronic, and then the 3.5 days. And again, you can see there just from those images that quite clearly a larger number of parasites in the acute stage are in S phase during the labeling procedure. But the final sort of data I just want to show you is that the infection of host cells, even during the chronic stage, is not long term. So despite the fact that, well, let me talk you through the experiment first. This is a pulse chase experiment. We labeled chronically infected mice as before, two injections, and then sacrificed the mice 1 day, 7 days, or 14 days after the labeling. And we then went to see how many of the parasites are still labeled at that period. And as we expected, there's a decrease over time. In the chronic stage, again, as before, we get around about 20% of parasites are infected, and then there's a steady reduction. So by day 14, there are very, very few labeled parasites. And that can be shown clear on these images here. So here we have our 1 day infection as before. Here we have, again, looking at the labeling 7 days after EDU labeling and 14 days. And we've illustrated the data down here. So you can see that after 7 days, very few of the cells are now labeled with EDU. So any cells that were originally labeled, presumably, as the parasites replicate, the EDU labeling gets less and less intense, and within 2 to 5 generations, it disappears. However, with the 14-day chase here, we analyzed almost 90 cells from several infected mice, and we only found one infected cell where we could still see EDU labeling, and that's shown here. And this is a normal pattern where we no longer see EDU labeling. So that tells us that there are very few cells that survive, infected cells that survive full 14 days, because none of the parasites that were infected at the end of the experiment, only one out of this 90 still had parasites that were EDU positive. So this suggests that long-term infections in the chronic stage are not how the parasites survive. They undergo the normal or maybe elongated rounds of infection, replication, and host cell lysis. And we are not really able to identify any cell types that we could define as dormant in this particular context. Okay, just to summarize that, parasites are restricted to the colon by immune pressure, and typically the colon contains only a few hundred parasites. The majority of these are in the circular muscle layer, in smooth muscle cells, and the majority of the parasites are in host cells that contain very large number of parasites, greater than 200. The replication rate appears slower in the chronic stage than the acute stage, and during the acute stage, infection of individual cells does not appear to be long-term. So that's not really an explanation of why you get this lifelong infection. Simply that parasites are sitting there for extended periods. That doesn't seem to be the case. Okay, so let's still get, and just in the final couple of slides now, get back to the original question. Why does T. cruzi cause Chagas disease? Okay, so 3 or 4 years ago, myself and Michael Lewis wrote a review for Trends in Parasitology in which we hypothesized a model for the progression of Chagas disease in specifically the bulb C mouse. So these are the organs in a naive mouse. You get an infection. It's pantropic. All of the organs are infected in the acute stage. But once we get to the chronic stage, a very robust T-cell mediated response, mainly CD8, that reduces the parasite burden by 100 to 1,000-fold, restricts parasites, in this particular model, predominantly to the colon and the stomach, and rarely in other organs and tissues. However, if you look at this model, then I think you can see a problem here for the parasite. How does it get transmitted? the triatomine bug will certainly not have access to the colon or the stomach, and therefore the parasite must have a strategy for getting to the periphery of the mouse to allow transmission to occur during a blood meal. And so what we hypothesized then was that intermittent trafficking of infected cells or parasites from the reservoir sites, be they in the colon, the gut, or in other mice, maybe the skeletal muscle. And these parasites then infect a large number of organs, including the skin. I showed you earlier what the skin looks like. What we don't know is whether the parasites we see in the skin are localized there on a permanent basis, or whether they are constantly trafficked from other reservoir sites and occur only transiently in the skin. That's something that we've got to address. But it's implicit in this model that some of the parasites will reach the heart and other organs on a transient basis. But as soon as you get to these other sites where there is a strong inflammatory response occurring when the parasite gets to that point, these small localized and transient infections of the heart will generate a rapid inflammatory response that will ultimately result in tissue damage, fibrosis, and localized collateral damage. So what we then will predict that over time, this collateral damage will accumulate, and it's this accumulation of collateral damage that leads to the time-dependent pathology that is associated with Chagas disease. So what we're saying is that cardiac pathology is driven by periodic trafficking of parasites into the heart at a frequency that is determined by host and parasite genetics and perhaps by environmental factors. Okay, so let me just finish now on a couple more slides, because it's implicit from this model that early curative treatment should block cardiac pathology, because if you can stop the long-term trafficking of parasites, if you can treat people early in infection, you should block the accumulation of pathology. So the experiment we designed to demonstrate this, again, other people have carried out similar experiments to demonstrate this, but I'll just show you our data. And what we had here was cohorts of mice that were infected, and then they were treated at 14, 22, 66, and 110 days. And they were treated with benzonidazole for 20 days. And here we have some images. Here's the mice that were treated, initiated at day 14. You see that by day 60 and day 168, they're completely clear of parasites. And similarly with those that are treated at day 66, 110, or day 22. We found that in this mouse model, benzonidazole at this concentration for that length of time invariably leads to sterile cure. Now you can see here the endpoint of this experiment was between 156 and 169 days. That wasn't out of choice. It was simply, we predicted we need very large cohorts of animals because of the spectrum of pathology we expect to find, and indeed that was the case. So we used very large numbers of mice to give our data sufficient statistical robustness to try and draw some conclusions. And that's shown on the image here, and I'll just go through that slowly. So here we have collagen that's laid down, so that's a proxy for fibrosis. Here we have non-treated cells, and you see it. This is essentially background collagen here. Here we have non-infected cells that were drug treated, again, just to demonstrate that the drug itself doesn't confer fibrosis. And here we have an infected mouse that was not drug cured. And you can see here there's a wide spectrum of pathology as judged by fibrosis. And actually this mirrors very closely what we see in human patients, where there is a wide spectrum of pathology ranging from nothing significant to quite severe damage. But what you can see here is in the mice that were treated when treatment is initiated at day 14 and day 22, that there was no subsequent pathology when we examined the mice at the experimental endpoint, approximately 6 months after the infection. However, where treatment was delayed for longer periods, and for 66 and especially for 110 days, you see that when we examined these mice, then presumably pathology had already developed before the mice were drug treated. Drug treatment will not affect the fibrosis, and these mice had levels of fibrosis very similar to those that were non-drug treated. So this suggests that if we want to prevent pathology in human patients, and again that's extrapolating from mice, then we should aim to treat people as early as possible. So the further key questions are, and ones that we and others are looking at, is why is T. cruzi able to survive as a lifelong infection? What is it about the GI tract in some mice or the skeletal muscle that despite there being a very robust T-cell mediated response, the parasites can survive for months in terms of the mouse and years in terms of humans? Is it something about the parasite? Is there a role for the reduced parasite proliferation that was hinted at from our experiments? Is there a role for what's being called dormancy in the ability of the parasite to survive? These are really important questions, both from understanding the disease and from developing new drugs. And again, finally, it's as a community, I think we've got to aim at improving the drugs and diagnostics so that we can roll out better treatment and access to treatment for those that are infected, because certainly I think all of the experiments from ourselves and others now are pointing to the situation where if we treat infected people as soon as possible after they're infected, we will alleviate the development of subsequent pathology, which again in humans takes many years to develop. So it does give us a window of treatment in the case of human disease. Okay, so I think I'm just about gone my time now. Just to acknowledge all of the T. cruzi group at London School. Again, these are the people who have done virtually all of the work that I've presented. They're a really great bunch of people. And again, if there are any people wanting to come to visit the lab and join us for a few months, where once the pandemic is over, we'll be very happy to receive you. And again, I'll just briefly acknowledge many of our collaborators, not all of whom were involved in this aspect of the work. Okay.
Professor John Kelly, thank you very much for this beautiful presentation, like an amazing work of cell biology and parasitology. Can you hear me?
Yes, very well. Yes.
Okay, just to be sure. So we have a lot of questions coming, and hopefully we have some time for you to address some of those. Would that be okay?
Yes.
All right. Okay, so we have a lot of people asking about the foci of infections and the dynamics of the infections you have been showing during the presentation.
Yeah.
So for example, Jose Raj was asking if you know why is the specificity of the location of Trypanosoma cruzi in the circular muscle layer. And Viviane Valença, maybe you can join this too, was asking what determines the migration of the parasite? Is there any kind of molecular attractant in the tissue that could define that?
Okay, again, these are really interesting questions. In terms of the location of the parasites, I think there are two obvious determinants of that, and one will be the immunological microenvironment of the parasites and their ability to survive in that situation. And secondly, might be at a biochemical level in the environment of smooth muscle, which are the types of cells that best support parasite proliferation. In terms of how the parasites are trafficked, I don't know if there's any chemical attractant that is involved in the process, or whether it's largely a stochastic and random process. My feeling is towards the latter, that the parasites don't necessarily migrate, but I have no evidence for that. It's just knowing the sort of the nature of T. cruzi and the way it replicates, and that's my feeling. So nothing further to that.
Thank you very much. So
okay.
As you're discussing about replication, Carolina Keller was asking, you showed that the chronic stage, the parasite replicates a little bit less, but okay, I just lost it. Okay, so but they make big nests inside with a lot of parasites. So do you know if there are any parasites dormant in those stages, and if there are, could they eventually spread and cause an acute infection again?
Okay, again, that's a good question. Obviously, within these huge nests, it's very difficult to identify if a parasite is non-replicating. Certainly, we using the EDU labeling approach, that wouldn't necessarily tell us whether a parasite is dormant or replicating, because the EDU labeling is simply a narrow, sharp snapshot to identify parasites replicating at a specific point in time. What we didn't find in the chronic stage certainly was, in any significant number, parasites that were labeled at the beginning, that's on day 1, and then 14 days later, there was no sign of any parasites that had survived that long apart from one infected cell. So if there is a dormancy involved, either we haven't detected it yet at any great level, or possibly it's something that is quite a rare event, and is certainly in the colon. It may well be a rare event that its role in the life cycle of the parasite, its role in the development of disease, again, is an interesting question, and it's one that we and others are looking at.
I see. Thank you very much. So talking about a little bit about the temporal dynamics that you are showing, especially like during the chronic stage, do you know if the temporal dynamic of the immune system also has a role on determining that? And a question for myself also, if I could add into that, is, have you checked if there is any kind of circadian rhythm in the parasite dynamics? So are these two things cross-talking?
Okay, I didn't hear the question too well, but I'll try and answer. Obviously, the dynamics of the infection are, I presume, down to the interaction between the parasite and the host immune system. But we know from Rick Tarleton's work that the CD8 response doesn't undergo exhaustion. So there's still, throughout the course of infection, a very dynamic immune response. And certainly parasites are rapidly killed if you inject a mouse that it is immune, or either through vaccination or through previous infection. We find that the vast majority of parasites are very rapidly killed. So there is a functioning immune response. Why that doesn't function in the context of these sort of what we might define as reservoir sites is something that we don't know. And again, it's something that is an object of our current research. I didn't catch the last. Could you ask me the last question again?
Sure. Yeah. So, do you think that there is any kind of circadian rhythm pattern from the host that might be affecting the parasite dynamics?
I have no evidence to support or otherwise a circadian rhythm. Yeah, I don't know. I have no evidence, and it's not something we've looked at.
Thank you. Some people were asking about the importance of the genetic background. So I believe you started presenting with, and you showed data from 2 different genetic backgrounds. And then you're showing a lot of data, but I was not sure about which genetic background you are talking about. And also, some people were asking, could the difference of heterogeneity of the genetic background in the strain you're working with explain the fact that some small amounts of cells carry a lot of parasites? Maybe those are highly efficiently replicating parasites?
Okay, yeah, I should have been more definitive. The vast majority of experiments that I presented today were with bulb C mice, and I did indicate where we've used C57 black or C3H. Now, most of our work in the lab involves either bulb C or C3H strains of mice. As I mentioned in the talk there, in the bulb C, the parasites are restricted more specifically to the GI tract, and you see less dissemination. In the C3H mice, again, there appears to be less restriction. Again, it's down to obviously the genetic background of the mouse, although we can't define what the parameters are. And the more wider dissemination in the C3H mouse is reflected in greater fibrosis. And we also find that some mice do die of cardiac arrest in the case of C3H, but very, very rarely in the case of bulb C mice. Now, again, we're looking here at this interplay between the host and the parasite genetics in terms of the disease pathology, in terms of how the parasite survives in various backgrounds. And again, this is going to be quite a wide range of interplay in different cell types, perhaps, and obviously in different mouse models. I think we are very realistic about using mouse models, and that they are models, and models always have the limitations. So all we can take with mouse models can teach us things, but they can't necessarily give us a full picture of what happens in a human patient. But I think with the mouse models that we've used, in terms of pathology, they mirror quite many of the issues in humans. And similarly, in drug susceptibility, we find that mice are good models for what happens in humans if you back translate clinical trials into mouse models. So there is a lot of interaction at the immune level, obviously influenced by the genetics, and we're still at a very early stage in trying to dissect the precise mechanisms that are involved in these processes. So I apologize if a lot of the work is largely descriptive at this stage, but it does give us now a framework to go down a more mechanistic route.
Thank you. But what about the genetic background of the strains from the parasites you're working with?
Okay.
Not only the mice.
We've made bioluminescent strains from most, covering virtually all of the different parasite lineages. That's the discrete typing units, DTU 1 to 6. And we find that there is variation in the extent of infectivity, but at the moment, it appears to be almost as much difference between lineages as within lineages. So we haven't really got anything definitive to say about whether one lineage has different properties to another lineage. It's simply because there is such a diversity in the way that the parasites infect mice. And again, we've also got to be careful dealing with parasites that have been in culture. How have they adapted to culture, and how has that affected infectivity? But all I can say is that the infection profiles that we see are very, very reproducible within each strain. And in general, the profile of seeing an acute stage generally 14 to 21 days, followed by a gradual transition to the chronic stage, is something that is reproducible in all the strains.
Okay, amazing. So I have a couple more questions from the YouTube and from Google Meet, and maybe later we could open up for a couple of questions from if someone wants to speak themselves. Hopefully that would be okay. And Professor Antonio Carvalho was asking if you had the chance to perform any functional heart analysis in your mice that did not show parasites during the chronic infection.
Could you repeat? I didn't catch that one too well.
Sure. I apologize. So yeah, Professor Antonio Carvalho was asking if you had a chance to perform functional heart analysis in the mice that did not show parasites during the chronic infection.
No, it's not. Apart from looking at the pathology in terms of fibrosis and a number of other stainings, that's not something we've followed up. But I should emphasize that what we hypothesize is that in all cases, the heart of mice will be infected. It's not a persistent infection. And in some strains of mouse, the cardiac infection appears to be more frequent than in other strains. But again, what we're saying is that if we take mice and do the ex vivo analysis and the heart is negative for parasites, that doesn't necessarily mean that it was negative yesterday or last week. It just means it's negative at the point of sacrifice. So these are snapshots that give us an indication of the temporal nature of infections. And it's this sort of constant rounds of reinfection, we believe, that gives rise to the pathology. So the parasite is very much involved in the pathology that's associated with cardiac damage.
Thank you. And related to pharmacology and treatments, there are two questions from YouTube. So Jadel Kratz was asking if you believe that slow application in the chronic stage represents a big barrier for antiparasitic efficiency at this stage of infection. Do you have any comments on that?
Okay, so again, this goes back to the concept of dormancy and slow replication. So I would think that certainly slow replication, intuitively you would think that if a parasite is replicating more slowly, then if you have a drug that acts by inhibiting metabolic pathways, then may be less sensitive to that drug. But I don't think it's necessarily a barrier. I think it's just what we have to do is better understand the biology of the parasite and see how the various proliferative states of the parasite then impinge on sensitivity to different classes of drug. So it's the proliferation rate can have an impact, but we don't really have a sufficient understanding of the nature of this in the chronic stage of the infection to be too certain at the minute. But it's certainly something that has to be built into drug development strategies, is to better understand what's going on.
I see. Professor Vanderlei de Souza was asking which kind of instruments do you use for bioluminescence imaging? And he was asking if you try to couple that with X-ray tomography.
We haven't tried to do that. We use this. We use the Spectrum instrument, which unfortunately actually broke down today. We're hoping to get it fixed on Monday, but it's a very good instrument, and it's very rare for us to have any problems with it.
I see. I don't know if anyone from Google Meet would have interest to ask the questions themselves.
I have an interest.
Okay, Julie, please go ahead.
Okay. I'm sorry, John, but I lost contact with you, so I don't know if you have already answered the questions that I would like to raise.
So is this, who am I speaking to?
This is Julio Schachter.
Oh, hi, Julio. Hi.
Hi,
Go ahead.
John. Yeah, it was very stimulating, your talk. There are many aspects that I didn't have the chance to see in my last visit in London. But there is one interesting question related to the fact that you only have a few cells that are over-infected during the chronic stage. What happens if you simply deplete the T cell response at that stage? You know, how much of the pressure on the infected cells is actually due to the fact that you have an ongoing T cell response? Is this an intrinsic effect that they have to do with a different adaptation? Or even the possibility that I think it would be interesting to discuss, that you have a little bit of fibrosis there, so you don't have sufficient density of microvessels in order to support the widespread growth that you see in the acute phase. Yeah. Because as you may remember, we are working very intensively on that aspect in the model in the cheek pouch. And we have many, many interesting features that nicely fit to what you have described, including the fact that during the early stages of infection, you have a very, very delicate, a subtle leakage of plasma, which helps the angiogenesis that is full-blown when you have the egress of the parasites with cell death. So, I mean, if you compare this scenery with the chronic stage, in the chronic stage, maybe there are structural differences and biological differences that don't give an opportunity to have a nice angiogenic process, you know, to develop. So what the contrast that you see may be at the end result of, you know, the pathology itself. But I think that the microcirculation could have a very crucial role, including during the transition to the heart, because as we have discussed in London, you may have a very intense edema, which is very, you know, short time. It's very short-lived. And during that edema, you have conditions for the fibrosis to develop, especially because maybe you don't have angiogenesis as you should have in order to have a nice repair of the tissue. So I was thrilled with your presentation. I could discuss with you maybe another 15 questions, but this is basically what I would like to leave for you. Okay, I mean, you're asking questions, Julio, that are a bit outside my specific area of expertise, and it's obviously this is an area where you are a leading researcher, and I think any ideas that you can come up with that can correlate between the type of data we have and the type of analysis that you're doing would be very welcome. Just to mention back to these large meganests that we find in the colon, again, just to remember that although a big percentage of the parasites are in what is essentially one or several meganests, you have to remember that there are many more infected cells that contain relatively small numbers of parasites. It can vary from 1 to 20. Sure. Yeah. No, that makes sense. Yeah.
Okay.
But you didn't answer the question about the adaptive immunity. How much of the contrast is actually due to the adaptive response?
Yeah. All I can say on that, Julio, is that these are the type of experiments we are doing at the moment.
Okay.
And we hope to get a clearer picture within the next few weeks or months.
Yeah, but I think that you could also consider doing the adaptive transfer of the T cells from the chronic mice, or even using, you know, the protocol that you have used from Tarleton, you know, the benzodiazole treatment and transfer. Because even in the acute phase, if you do the assay with the NDP and now look at the distribution of the parasites, maybe you could actually recapitulate the conditions that you see in the chronic phase. So, that should not be such a difficult experiment to do. I think it would be useful.
Okay, thanks for the advice. That's good.
Okay, Professor John Kelly, thank you very much for your time. I guess I will now pass to Susanna Fraser, that will start wrapping up this presentation, this talk. So Susanna, do you want to go ahead?
Oh, thank you very much, Professor Kelly, for your excellent talk. In the name of all the students for our graduate program in biophysics, from the Carlos Chagas Filho Institute of Biophysics from the Federal University of Rio de Janeiro, it was a pleasure to have you with us today and have the pleasure of hearing about your excellent work. Thank you very much.
Thank you for listening.
Professor Vanderlei.
Well, I think that we had the opportunity to hear a very nice talk. John established a new model of work. And of course, with this bioimage model, coupled to immunofluorescence microscopy of the sections, you can really contribute to a better knowledge of what is going on in the animals. I have one last question. Did you observe mega colon or mega esophagus in any of your chronic mice?
Yeah, we are focused mainly on cardiac pathology and infections of the colon. Michael Lewis, whom I'm sure you know, is currently working on the pathology associated with the GI tract. And so Michael's work's currently ongoing, and I'm sure if he'll be very happy to discuss his work further with yourself.
Okay, John, I would like to thank you very much for your talk.
Thank you.
And I hope to see you personally in a few months. Okay.
Good.
Thank you.
Discuss his work further with yourself.
Okay, guys, thank you very much for being here with us today. Thank you.
Okay, John,
again. And hopefully
I would like to thank you very much for your talk. And I hope to see you personally in a few months. Okay.
Good.
Thank you.
Discuss his work further with yourself.
Okay, guys, thank you very much for being here with us today. Thank you.
Okay, John,
again. And hopefully
I would like to thank you very much for your talk. And I hope to see you personally in a few months. Okay.
Good.
Thank you.
Foi tão difícil, não
Discuss his work further with
Okay,
yourself.
guys, thank you very much for being here with us today. Thank you.